Welcome to a new lecture. I hope you are all doing well. It's our lecture 5 and in this lecture we will discuss about enzymes. So the main objectives of today's lecture. In this lecture we will discuss about catalysts, characteristics of enzymes, mechanism of enzymes action, and effect of change in temperature and pH on the activity of enzymes. So these are the objectives of today's lecture. What are enzymes? Enzymes are the most important group of proteins which are biologically active. Enzymes act as biocatalysts. What is the term biocatalyst? So if we describe the catalyst, then we can say that catalyst is a substance that can be added to a reaction to increase the reaction rate without getting consumed in the process. So catalysts are the substances that increase the reaction rate but they are not consumed in the process. So they speed up a reaction by reducing the activation energy or changing the reaction mechanism. What is activation energy? Activation energy is the minimum amount of energy that is required to start a reaction. We will describe the activation energy in detail in further slides. So enzymes are known as biocatalysts because they speed up and regulate the biochemical reactions and are not changed during the reactions. That's why enzymes are known as biocatalysts. What is substrate and products? The molecules at which enzymes act are called substrate. So substrates are the molecules on which the enzymes act. And enzymes convert them into different molecules which are known as products. Enzymes are composed of hundreds of amino acids joined together and coiled upon themselves to form a globular structure. What is active site? Only a small portion of enzyme molecule is directly involved in catalysis. This catalytic region is known as active site. What is the function of active site? Active site recognizes and binds the substrate and then carries out the reaction. As we have described earlier that reactants are called the substrate and the substrate are attached to the active site consisting of only a few amino acids. So active sites are those sites on which substrates are attached and then the reaction proceeds. So in this diagram we can see that here is an enzyme and it is the substrate and this site is called the active site of the enzyme. So substrate attached to the enzyme to its active site and here structure is developed and then this is further break down into products. So here it is the product A and B. What is activation energy? All chemical reactions require activation energy. Activation energy can be defined as minimum energy required to start a reaction. So it is the minimum amount of energy that is required to start a reaction. Enzymes lower the activation energy in several ways. They may alter the shape or change the distribution of substrate and reduce the requirement of energy for this change. So enzyme lower the activation energy. They may change the shape or charge distribution of substrate. So we can say that in the presence of enzymes, reaction proceeds at a faster rate, and without the enzymes, the reaction would proceed at a very slow speed and it will make the life impossible. So as we know that enzymes lower the activation energy. So here is the activation energy without the enzymes, and it is the higher activation energy without enzymes. So when we use the enzyme, it lowers the activation energy. So it helps to speed up a reaction. What is intracellular enzymes? Intracellular enzymes are those enzymes that function within the cells. For example, enzymes of glycolysis working in the cytoplasm, and these are known as intracellular enzymes. Here is another term that is known as extracellular enzymes. What are extracellular enzymes? Those enzymes that function outside the cells are known as extracellular enzymes. For example, pepsin enzyme working in the stomach cavity is an example of extracellular enzymes. What is cofactor? Some proteins consist solely of proteins. Some enzymes consist solely of proteins. Others also have a non-protein part known as cofactor. So cofactor is a non-protein part. What are the functions of cofactor? It is essential for the proper functioning of enzymes. The cofactors usually act as bridge between the enzyme and its substrate, and they contribute directly to the chemical reactions which bring about catalysis. Sometimes cofactors also provide a source of energy and drive the reactions which would otherwise be difficult or impossible. So cofactor can be inorganic or organic. So inorganic cofactors include metal ions like magnesium, copper, zinc and others, while the organic include flavin and heme. Here we can see that this is a cofactor and it is an inactive protein. So when cofactor binds to it, it activates the protein. That's why it helps for the proper functioning of enzymes. What is prosthetic group? If the non-protein part, what is non-protein part of the enzyme? It is known as cofactor, which we have described earlier. If the non-protein part or organic cofactor is covalently bonded to an enzyme, it is known as prosthetic group. Covalently bonded means tightly bonded. What is coenzyme? If the non-protein part or organic factor is loosely attached with enzyme, it is known as coenzyme. So if it is covalently bonded, it will known as prosthetic group. But if it is loosely attached with the enzyme, it is known as coenzyme. Some important vitamins like riboflavin, thiamine and folic acid act as coenzymes. What are the main characteristics of enzymes? All enzymes are globular proteins. They increase the rate of reaction without themselves being used up. Their presence does not affect the nature or properties of end products. Small amount of an enzyme can accelerate the chemical reactions. Enzymes are very specific in their action. Enzymes are sensitive to even a minor change in pH, temperature and substrate concentration. Some enzymes require cofactor for their proper functioning. Enzymes lower the activation energy of the reactions. What is the mechanism of enzyme action? When enzyme attach with the substrate, a temporary enzyme substrate complex is formed, which is known as ES. ES is the enzyme substrate complex. Enzyme catalyzes the reaction and substrate is transformed into product. So after this, enzyme substrate complex breaks and enzymes and products are released. As we can see that here, enzyme combined with its substrate through its active site and then enzyme substrate complex is formed. Then enzyme catalyze the reaction and substrate is converted into products. Then enzyme substrate complex breaks down and it is converted into enzyme plus product. So here we have two models which describe the mechanism of enzyme action. First was the lock and key model. What is lock and key model? So a German chemist Emil Fischer in 1894 proposed the lock and key model. According to this model, both enzymes and substrate possess specific shapes that fit exactly into one another. This model explains enzyme specificity. So according to the lock and key model, enzymes and substrate possess specific shape and they fit into one another. According to the lock and key model, substrate possesses a specific shape that fits into a specific enzyme. So then enzyme substrate complex is formed and then the further reaction proceeds. But then a new model was introduced, that was the induced-fit model. In 1958, an American biologist, Daniel Koshland, gave a modification to lock and key model and he proposed induced-fit model. According to this model, active site is not rigid structure. It is molded into the required shape to perform its functions. So now induced-fit model is more acceptable than lock and key model. So according to the induced-fit model, active site is not rigid. So it will take the shape of the substrate. It changes its shape according to the shape of the substrate. So here we can see that the substrate has different shape and the active site is the different shape. But then we can see that the active site changes its shape according to the shape of substrate, and then enzyme substrate complex is formed. So the major difference between induced fit model and lock and key model is that lock and key model described that active site is rigid, but according to the induced fit model, active site is not rigid, but it molded into the form of this shape of the substrate. What is specificity of enzymes? There are over 2000 known enzymes, each of which is involved in one specific chemical reaction. Enzymes are also substrate specific. So a specific type of substrate will attach to a specific enzyme. For example, the enzyme protease, which breaks peptide bonds in the proteins, will not work on starch, which is breakdown by an enzyme amylase. Another example is of lipase enzyme that acts only on lipids and digests them into fatty acids and glycerol. So, specificity of the enzyme is determined by the shapes of their active site. Active site possesses different geometric shapes that fit with specific substrate. Here is the substrate 1, substrate 2 and substrate 3, and the shape of the active site is this. So a specific type of substrate will attach to the enzyme. What are the effects of changes in temperature on the enzyme activity? Increase in the temperature will speed up the rate of enzymes catalyzed reaction, but only up to a point. Every enzyme works at its maximum rate at a specific temperature, which is known as optimum temperature for that enzyme. So the optimum temperature for the human enzymes is 37 degrees Celsius. When temperature rises to a certain limit, heat adds in the activation energy and also provides the kinetic energy for the reaction. So reactions are accelerated. But sometimes when temperature is raised above the optimum temperature, heat energy increases the vibration of atoms of enzymes and the globular structure of enzyme is lost. This is known as denaturation of enzymes. So in this diagram we can see that here the temperature is increasing and enzymes gains more kinetic energy and rate of reaction is accelerated. At optimum temperature, which is the 37 degree Celsius, the reaction will be in highest speed. But after this, when the temperature is raised above the optimum temperature, which is 37, when temperature is raised more than 37, then heat increases the vibration of atoms of enzyme and the globular structure of enzyme is destroyed, which is known as enzyme denaturation. It results in a decrease in the rate of enzyme action and it may be blocked completely. What are effects of changes in pH on the enzyme activity? All enzymes work at their maximum rate at a narrow range of pH known as optimum pH. If there is a change in the pH, it can cause the retardation in enzyme activity or can block it completely. So every enzyme has its specific optimum pH value. For example, pepsin, which works in stomach, is active in acidic medium which have low pH, while trypsin, which works in small intestine, is active in alkaline medium that have high pH. So change in pH can affect the ionization of amino acids at the active site. In this diagram we can see that at optimum temperature the rate of enzyme reaction increases. I hope this lecture helped you to understand the topic. Thank you for listening.